Neuroplasticity, the brain that changes itself. What are neurons? Neurons are the basic units of the brain and nervous system, the cells responsible for receiving messages, sending motor commands to our muscles, and transforming and relaying the electrical signals to our body. More than that, their interaction define who we are as people. Neurons are nervous system cells that communicate information to the nerve, muscle, and gland cells. Axons, dendrites, and a cell body make up the majority of the neurons. The nucleus and cytoplasm are found inside the cell body.
The cell body's axon spreads from there and frequently divides into several smaller branches before reaching the nerve terminals. In order to communicate with other neurons, dendrites extend from the cell body of the neurons. Synapses are the places of contact where two neurons can interact. Synapses formed by the tips of other neurons' axons cover the dendrites.
There are different types of neurons, both in the brain and the spinal cord. They are divided according to where they originate, where they project to, and which neurotransmitters they use. What is neuroplasticity? Neuroplasticity refers to the physiological alterations to the brain brought by our interactions with the outside world. The connections between the cells in our brains remodel from the moment that the brain starts to grow in utero until the day we die in response to our changing demands. We may learn from and adjust to various situations, and thanks to this dynamic process, neuro refers to neurons, which are the nerve cells that make up the brain and nervous system. Plasticity relates to the brain's malleability, which is referred to as being easily affected, trained, or controlled. Neuroplasticity is the ability of nerve cells to adapt or change.
There are two types of neuroplasticity: functional plasticity and structural plasticity. Functional plasticity refers to the brain's capacity to transfer functions from one injured area to another unharmed area, while structural plasticity refers to the brain's capacity to truly alter its physical makeup as a result of learning. How neuroplasticity works. An estimated 2,500 synapses or tiny spaces between neurons where nerve impulses are conveyed exists in each cerebral cortex cell at birth. By the time a child is 3 years old, there are astounding 15,000 synapses per neuron. However, the average adult only has roughly half as many synapses. Why? Because some relationships get stronger as we acquire new experiences, while others go away. Synaptic pruning is the term for this procedure.
Stronger connections form between regularly utilized neurons. Those that are utilized little or never pass away gradually. The brain may adjust to a changing environment by creating new connections and removing weak ones. Benefits of neuroplasticity. The neuroplasticity of the brain has several advantages. Having an adaptable and flexible brain supports the capacity to pick up new skills, the capacity to improve current cognitive capacities, stroke and traumatic brain injury recovery, bolstering places where function has decreased or was reduced, and enhancements to improve mental fitness. The limits of neuroplasticity. Neuroplasticity is only capable of so much. Animals other than humans exhibit extensive brain plasticity. They are unable to acquire a human language or master complex mathematical operations because of the limited capacity of their brains. Neuroplasticity just modifies the precise neural substrate of a cognitive function or partially modifies a brain function for a season, since it relies on physiologically accessible material.
The phenomena of neuroplasticity. It is possible for changes to be spatial or temporal. Long-term and short-term changes in temporal relationships are further separated. Furthermore, there is a spatial alteration at synapses, inside neurons or inside glial cells. Individual neurons also undergo modifications in functionality. A single neuron experiences the following processes: excitatory of postsynaptic potential, synaptic strengthening, long-term potentiation, and long-term depression. The population of neurons is also affected by structural changes. The brain can change itself through things like unmasking. When two events happen at the same time, those neurons involved in the experience fire together, thus they associate themselves with each other. As this connection becomes stronger, these neurons wire together.
Scientists thought for a long time that each area of the brain had a distinct function, and if that area got damaged, there was no getting that part back. Though some areas are responsible for specific roles, many often overlap and help one another. If one pathway becomes blocked, a secondary one is exposed and used instead. This new pathway grows stronger with repeated use in a process called unmasking. The experience of Cheryl Skills is used to illustrate this phenomena. She lacked a working vestibular apparatus, the organ that helps us to balance, which caused her to have balance issues for years. Paul, a pioneer in the field of neuroplasticity, invented a gadget called an accelerometer that sends balance data via electrode on her tongue.
With our simple imagination, we can alter our brain. The brain in the body may physically alter just by visualizing what we desire. Patients who suffer phantom pain are one illustration of this. This is the condition where the person who have lost a limb feel the pain that originates from the missing limb. Phantom pain, once thought to be a psychiatric issue, is now understood to be a nervous system issue. The brain map for the missing limb is eager for information and send growth factors to neighboring neurons. According to the explanation, explaining why the pain still occurs.
V.S. Ramachandran, a neuroplasticity researcher, developed a mirror box that displayed the mirror image of the functioning limb to trick the brain into thinking the missing limb was moving and responding to input. They might unlearn the phantom pain in this manner. It was extremely effective for a guy who had lost his arm in a motorbike accident and was feeling excruciating phantom pain. Visualization is another way imagination can alter our brains. There were two groups of beginning pianists in the experiment, while the other group spent the same amount of time practicing the sequence. The first group sat in front of the piano and imagined performing it.
Researchers found that only doing mental practice caused the same physical alterations in the participants' motor systems as those who really practiced when they mapped the subjects' brains. They possess almost the same talents, in other words. The brain performing an action is not all that different from imagining it. The capacity of the nervous system to alter its activity in response to internal or external stimuli by rearranging its structure, functions, or connections is known as neuroplasticity or brain plasticity. Synaptic plasticity, which is a general term for neurons' capacity to alter the intensity and effectiveness of synaptic transmission through a variety of activity-dependent processes, is a key characteristic of neurons.
It was previously thought that the brain stops growing in the first few years of life and that connections between neurons in the brain are only established during this critical period while children were still young. Research has found that children with blindness have increased connectivity and reorganized neurocircuits when compared with children without this condition. These connections, it was thought, would then stay in place as we aged. For this reason, it was thought that only the developing brain was plastic and capable of making new connections. This idea has led scientists to go believe that certain parts of adult brain cannot regenerate or make new connections, and the functions controlled by those parts of the brain are lost forever. Thank you for watching, and we hope that you learned something. See you again.